Alrighty. Thank you, and I've really been enjoying this symposium quite a bit. It thinks at a very different level, and certainly I think many of us, when we went home, we certainly thought about some of our science in different ways, and I think that's an objective of some of these symposiums, and certainly has done that with me. So what we're here for is to talk a little bit about organisms, or really a cell, the anatomy of a cell. My laboratory really focuses on the interactions between different organisms. But I think some of the concepts that we are trying to think about certainly adapt to cells or entire organisms. And so, closer to the microphone. I got a voice here somewhere. It's been so. But anyway, I think most of you will agree that if you want to create a good model of a cell or an organism, you need to know all the components that make up this particular cell. And so the molecules really are going to define how you're going to build a model. And quite frankly, I think we're not very good at even taking a basic inventory. And so the end goal, I think, what I would like to see as an experimentalist is sort of a Google map view of an organism and cell, and then be able to answer questions with it. And so if you compare that to, let's say, Google Earth or Google Maps, what we want to do is be able to see all the buildings. We want to be able to see the trees, the streets. The equivalent for that, for me, is the makeup, the building blocks of the cells or the molecules. And so that's what we're trying to do. And so before you're going to create really good biological maps, how do we create such an inventory? And for me, I take it as a second step, maybe more abstract, but can we create a searchable map of molecules? And for me, I'm really trying to understand what a lot of specialized metabolites are doing within different organisms. But this could be similar to what are different specialized metabolites doing within different organelles. And so what we do is we collect mass spectrometry data. And so we weigh how heavy the molecules are, and that's really our efforts that we do. The one nice feature about this is that mass spectrometry is sort of blind, whether this is a single cell, a mixture of organisms, or et cetera, et cetera. And so here's one of these examples where we show mass spectrometry data being collected on a single microbial cell population, basically a single microbial colony, and we're getting mass spec data. We can do this on my postdoc's finger, and again, you can get mass spec data. Now, just this doesn't give you any insight in terms of the molecules. One of the things that you can do by mass spectrometry is you can break the molecules apart in the gas phase. And so what you're going to do is all the masses that you saw, you're going to break them apart, and in principle, the chemistry dictates how these molecules fall apart. And can we use that to our advantage to begin creating some sort of a Google Map style view to be able to begin annotating and taking an inventory of the molecules. So we can break these molecules. You now get a barcode signature of these specific molecules. And in reality, you create several million of these in a typical experiment. So we have a series of barcodes. In reality, they show up to look like this instead. And what we have begun to do is we've begun to sort of think about, well, how do we make comparisons between these molecules? It's very common to do this in gene sequencing or with gene repositories like GenBank, where you compare your protein or molecule DNA sequences of interest, and you find some other matches to that, and you infer a function with respect to those correlations. We can begin to think about doing the same thing here. And so first, what we need to do is find some sort of correlation score, and that they really match up and align these different spectra. And of course, we can represent those as these little nodes. So in this case, these nodes don't represent genes. No, they represent molecule classes. And so there's no biological function inherently embedded within these different correlations that we see. But then, we get this hairball, and really most experimentalists, they step back and say, this is too much. We can't really deal with anything like this. But nonetheless, they sometimes try, and that's what my laboratory tries to do. And then you can apply different algorithms to sort of rearrange your data and sometimes make it visually more appealing and really begin to be able to pull out some of the chemistry that is embedded here. And so in this case, every single node, if you get a cluster of these nodes, that represents structural similarity. And so once you have an optimized map that you're satisfied with, that you think you can pull out some chemistry in essence, you can now begin to add different attributes. You can add activity data. Here we show the intact mass of the molecules that were fragmented, and you can see nice correlations. If there's often what you will see is that related masses cluster very nicely based on that barcode signature. Occasionally we'll see multiple masses correlating in different regions. And the sugars, for example, there's a large number of different masses of sugars that typically correlate in specific regions. But you can also add other attributes. So here we say bacteria A or bacteria B, but you can also do organelle A versus organelle B. So to me, that's no different at that stage. And so we can then zoom in sort of these kind of clusters. And this is where it becomes chemically informative to me. And what I like about it is it's agnostic to the type of molecule that's there. It doesn't matter whether it's a lipid, peptide, protein, DNA, or sugar. It's agnostic. And so the first thing that I can do is very quickly, I can already learn chemistry by just zooming in on this particular cluster. That's step one. Now, if you're lucky enough, and this is what you would do in a metabolomics experiment, if you're lucky enough, you can annotate one of these molecules and begin to say something more. And I pulled out 3 of these molecules from that cluster that I just showed you. And these turned out to be different lipids that exist within my sample. And so we have different fatty acid chain lengths that are represented by these different particular nodes. And so one of the things about, I've heard a lot about multiscalar biology, and I think one of the common languages, if you want to really cross the boundaries at multiple scales, is the chemistry. And so I just grabbed some random pictures in many ways, but you can take organisms and you can explain this at the chemical level. You can literally pack almost anything, a cellular, or even go to a single cell, or in this case it's a microbe eukaryotic cell interaction, or at the organelle level. But the common language, again, are the molecules. And so if you really want to do, in my opinion, create a good model of a cell, you need to, or an organism, you need to be able to take good inventory of the molecules that are present. And so our approach is sort of this Google map approach that we've been taking here, shown on the right. I don't know if this is the best one, best way to visualize this type of data, and I would certainly welcome suggestions from the audience, certainly people who think more about visualizing data. But what this really allows you to do, once you can take a good inventory, then you can build better networks that are more adequately represented. You can now translate what some of these molecules are and translate that to human biology. For example, on the top we're showing we're interested in how microbiota, what kind of molecules they produce, and how that influences balance of health and disease. But we're also interested in microbe interactions. But again, it's the inventory of the molecules that allow us to begin translating the biology. And then finally, I want to leave you with the notion, well, Theodore will actually cover a lot more of these models that are created and later on. And finally, I want to leave you with the sort of acknowledgments to people that did a lot of the work in my laboratory, and I'm really grateful to have all those organisms in my lab. And then finally, I won't be here during the question and answer session, so I have probably 2 minutes left. So if there are any questions, I could probably take 1 or 2 questions before I have to leave. So thank you.